and getting sequences of tumor genes. And there was all this skepticism about, we knew, the sort of traditional look and think crowd had been doing, they hadn't been silent. They'd actually been doing a lot of important work defining key genes involved in cancer for a long time. And there was this feeling like, what would we really learn? Are we just going to spend a gazillion bucks to get the last 5% of information? That's what the skeptics felt, or would we actually learn something? But I think we learned quite a lot. I mean, we discovered a lot of new genes that cause cancer. One of the other revolutions that I've been fortunate to live through is to see, as we found specific genes that are suitable in cancer cells, people have been able to make drugs that specifically target those mutant genes. This is not a done deal. I mean, so you can develop resistance to those genes, but there are spectacular examples where we don't need to use the crude forces of conventional chemotherapy and can actually target a tumor and even wipe it out with a very specific drug that has minimal side effects. So that's not a promise that we can do that for everything, but it definitely has worked at least a few times. And from the cancer genome sequencing, we've got a whole bunch more of these type of things, and there's a lot of, I think, exciting activity in that area now. So I think we learned something, and we learned something that's going to be practical. But we'll see exactly how practical it is. We learned all sorts of things about how the genome changes when it's going from a normal cell into a tumor cell. And one of the things that excited me was a relatively recent discovery from not too far from here, the Sanger Center, by a guy named Peter Campbell. And sequencing cancer genomes, they found this really remarkable phenomenon where a single chromosome looks like it got sort of beat to hell, sort of smashed and kind of randomly stitched back together, with the rest of the genome being largely intact. And that was something totally unexpected that came purely from the actually reading the cancer genome like a book. You had to do that. You had to actually be sort of smart enough to piece it together so that you knew what you were looking at and who was abnormally connected to who. So we're learning. So from that, so he gave that a name called chromoversis, and the reason that that's so surprising is that we normally think of changes in the genomes or mutations as being kind of roughly a single number that would apply as a probability per base per generation. So it's, we kind of the very, very roughly say 1 in a million chance per base pair per generation. So the idea that this process, the chromosomes are all swimming around together, presumably in the nucleus, how you would actually do something highly local was really mysterious. And as it's turned out, from sequencing the cancer genome, there's all of this local phenomenon going on that we had no idea about, and we wouldn't have. So this, this is the disadvantage of looking. If you just look at the nucleus, the bag, the membrane bag that holds all the chromosomes, I mean, it basically sort of looks like a bag. And it would lead you to think that this would be sort of a well-mixed system, and everything would happen sort of, you wouldn't have a lot of local phenomenon within there, but we are, that's one of the really exciting cutting edge things, is that we're realizing that this bag called the nucleus actually has, places as different from Boston and Texas in it, they're worlds apart. So in the course of our work, studying what happened when you doubled the genome, we came upon a phenomenon where we realized that chromosomes, normally the chromosomes are in a mass, and then they're copied. You're getting ready to make two cells, right? So you have some chromosomes, you make a perfect copy, in principle, of all of them, and then they have to separate, and that's how you get two cells out of one. But we realized that there are ways in cancer cells in particular, but also in any cell that undergoes a genome doubling, that chromosomes can get caught in a kind of tug of war. And then we also realized that when the division of cell is this funny process that's a little bit like a child's game of musical chairs. And if a chromosome gets sort of left out in the cold, when the cell is finished dividing, it'll get trapped in its own little nucleus, which we call the micronucleus. So these things have actually been around and noticed to be important in cancer, or not known to be important. They've been described in cancer forever, but what they do is basically totally unknown. So, sort of in line with liking to go back to the older literature, we went through all this stuff and started wondering, well, maybe this would be a beautiful way to get a highly localized form of change in the genome if you just partition little bits off and they wind up in their own little structure. Then maybe bad things could happen to them, and then if they could reunite, then they could contribute, the bad things that happen to them in their separated space back into the rest of the genome. So, postdoc in my lab, Karen Krafta, did a series of experiments that showed just that the copying of the DNA in those things goes really poorly and leads to some kind of massive damage. So, and we know that those things can go back and contribute to the genome. So if we have a sort of a hypothesis that this might explain this local mutation phenomenon, something like this chromophore thing. So now we're doing a bunch of really interesting experiments. We've been in an era where scientists are sequencing what's there. They've sequenced different organisms. In cancer biology, they're just sequencing all sorts of tumors and knowing all the different regions of tumors, because they're a little different. One tumor is not homogeneous. You can have different regions that are all different. But I think we're kind of reaching the end of it. I've lived through several eras. I told you we had the Maximum-Gilbert sequencing, then we had the Sanger sequencing, and then there was massively parallel sequencing where it could really shotgun and sequence the human genome in a month or so. But now I think we're actually on the cusp of another revolution where we're going to use this sequencing the genome not as a descriptive tool, not just to look. So they learned a lot by looking and thinking, but I think now we're going to be able to do it as an experimental tool and try to recapitulate the evolution of this process in the laboratory and try to recreate, so our particular task, one of the projects in the lab, is to try to see if we can recapitulate this chromothripsis process in the lab, and then the readout then would be sequencing the genome. So, those are just, I think, some of the ways in which I hope you get a sense of how all of this information has sort of changed. It's been a fun ride. We're to like sailing on windy seas here, and all these things come along and pick us